Hello and welcome to Genius Science. This is the channel where we post all things GCSE science related. This video is a past paper walkthrough for the 2022 GCSE Combined Science Biology Paper 1 Foundation from the AQA exam board. So let's get into it. Alright, let's take a look at question number 1. So the foods are digested before they are absorbed into the blood, and here we have figure 1 showing us the organs in the human digestive system. And question 1.1 is asking us which organ is the stomach, and we have to tick one box. So by doing this you need to know the parts of the digestive system. So A is the mouth cavity, B the liver, C the stomach, D the small intestine, and this here would be the large intestine. So therefore our answer is C. Okay, question 1.2. What type of enzyme is produced in the stomach? And again, we need to tick just one box here. So carbohydrates digests carbohydrates and breaks them down into glucose. That doesn't happen in the stomach. Lipase enzymes, they digest lipids into fatty acids and glycerol. That does not happen in the stomach, happens in the pancreas, and carbohydrates being broken down is actually in the mouth. That leaves protease enzymes that break down proteins into amino acids. That does happen in the stomach, so that is your correct answer. Question 1.3 now. Again, all along the theme of the digestive system. Which term describes the pH in the stomach? And we have to give one reason why the stomach has this pH, 2 marks. So we're getting 1 mark here for the tick box and 1 mark here for the reason. So tick in 1 box. Okay, the pH in the stomach is acidic. And the reason for this acidity is the stomach produces hydrochloric acid. You can also say that it's the optimum condition, the best conditions for the protease enzymes to act. And you can also say that it produces hydrochloric acid in order to kill microorganisms. Okay, question 1.4 now. Which organ produces bile? And again, we're ticking 1 box. The liver produces bile. Just remember that. The liver produces bile. Okay, question 1.5 now. How does bile help in the digestion of foods? By saying help here, they're talking about the function. So the job of bile. And again, we just got to tick 1 box. We've got several options. And option 1, it increases the surface area of the fats. It is an enzyme that digests protein, and it makes the pH in the small intestine acidic. It is not an enzyme. It doesn't do this. It increases the surface area of the fats. This is sometimes described as emulsifying the fats. By increasing the surface area, makes it much easier for the fats to be digested and broken down further. Question 1.6 now. And we have to draw, that's our command word this time, draw 1 line from each food molecule to the reagent used to test for the food molecule. So we're now looking at food tests. Reagent is essentially just the chemical. That's just what that means. And we've got 2 marks here. Let's go for it. So the food molecules we've got are protein, starch and sugar. And then we've got the reagents, Benedict solution, biuret reagent and iodine solution. So this is just a straight recall question. Proteins, you use the biuret reagent. Starch, it's iodine solution. And sugar, is the Benedict solution. Do not get confused if you ever have any longer answer questions on these. When they ask for the test, don't say Benedict solution, say Benedict's test. It's a common mistake. Okay, question 1.7 now. And the question is, give one safety precaution a student should take when using Benedict's solution. Now, there's a couple of things you could say. I mean, we'll go through both of them here. Now, when you're dealing with a chemical, the obvious one, wear your safety goggles to stop the chemical splashing up into your eyes. And you can say wear gloves as another answer as well, because there are some chemicals inside the Benedict solution that could irritate your hands. We are now on question 1.8, and table 1 shows the results for one food sample. So a test has been carried out. Several tests have been carried out here. We can see the Benedict's test, Biuret test, and the iodine test have been carried out. And we've got the colours, so the results of those tests. So Benedict's solution has gone red, the Biuret solution has gone blue, and the iodine solution has gone black. And which of these tests show positive results? And we have to tick one box. So just a bit of background here on the Benedict's test. If you get a positive result, the Benedict solution will go from blue to either green, orange yellow, or brick red. Now, good way of remembering this is the hotter the colour, the more concentrated the sample is with sugar. So green is lowest sugar, orange medium sugar, and red it'll be very high sugar. So we can see here we have a positive test. Now for Biuret, again, Biuret solution is blue. And if it is positive for protein, it will turn to purple, or it can also turn pinky colour. You can see here it's blue, so that is a negative result. And for the iodine test, iodine solution, you can say it's an orange or an orange brown colour. And it turns to blue black or black. You can't say blue, so you can say blue black or black when starch is present, and you can see it's turned black here, so it's a positive result. So the positive results are Benedict's and iodine, so that's your correct answer there. Okay, question 1.9 now. Starch molecules are not absorbed into the blood from the digestive system. Give one reason why. Now, the main reason why here is that starch molecules are too large to pass through the walls of the small intestine into the blood. Now, you could also sort of give an answer here that alludes to the fact that the starch needs to be broken down into smaller, more soluble molecules, i.e. needs to be broken down into glucose before it can diffuse through the walls of the small intestine into the blood. Question 2 now, and figure 2, that's here, shows a section through a leaf. Okay, so we're looking into the leaf. And this is the structure of the leaf as looking through if you looked under a microscope. And we've got the palisade layer labelled here. In question 2.1, we have to give one way that the palisade layer is adapted for photosynthesis. Now, remember, photosynthesis is using light energy, so energy from sunlight, and converting it into glucose, which is a chemical energy store. A little bit of background there. And the palisade layer here is made up of cells called, surprise, surprise, palisade cells. And these structures here, these little grey dots there, they are chloroplasts. And there's loads of them because the main function of the palisade cells is to carry out photosynthesis. So in terms of our answer, the adaptations, you could say that the palisade cells contain many chloroplasts and that's more than enough there for 1 mark. You could also say that the palisade cells are located near the top of the leaf, because if they're near the top of the leaf, they can get more sunlight. So this here would be the top of the leaf, and that there is the bottom of the leaf. So you could say that the palisade cells are located near the top of the leaf. On to question 2.2. So gases pass into and out of the leaf through small pores in the surface of the leaf. Small pores, they're just tiny holes. Tiny holes. And what are the small pores labelled X called? So what's their scientific name? Because you can't say tiny holes. And we have 3 options: guard cells, stomata, and xylem vessels. So X, they are called stomata. The singular, so one of these, you would call it a stoma. And the gas is passing into and out of the leaf. A little bit of background for you. So into the leaf, it's going to be the carbon dioxide. And out of the leaf, you're going to have some oxygen, as that's a product of photosynthesis. And you may also, due to transpiration and a bit of evaporation on the surface of the leaf, you might get a bit of water vapor leaving the leaf as well. So on to question 2.3, and the student viewed a section of a leaf using a microscope, and they measured the length of one of the palisade cells with the image being 28 millimeters, and it's been viewed at a magnification of times 400, so it's looking 400 times bigger than what it would be in real life. And the question here is asking us to calculate the real length of the palisade cell in millimeters, and using the equation here. So real length equals image length divided by magnification. So real length equals the image length divided by the magnification, which is 400. Okay, now you'll get a mark for doing the correct substitution step, even if your answer in the end is wrong. And if we carry out that calculation, we would end up with a real length of 0.07 millimeters. Now, here they're asking us to convert the real length of the cell. So sorry, this mark here, if you get the right answer here, that'll give you 2 marks. Real length, so back to this question, convert the real length of the cell from millimeters to micrometers. And here they're telling us that 1 millimeter equals 1,000 micrometers. So using our answer here, 0.07, we need to times it by 1,000, and that will give us a real length of 70 micrometers. Question 2.4 now. And carbon dioxide can move into and out of cells. What is the process by which carbon dioxide can move into and out of cells? And we've got to tick 1 box. Now, the answer here is diffusion. Gases always move into and out of cells via diffusion. Active transport would be something like mineral ions moving from the soil to the root hair cells. And osmosis only concerns water molecules. And while we're here, let's define diffusion, just so you know for your revision. It's the movement of particles from an area of high concentration to low concentration. And another key part of this definition, we would say, is the net movement of those particles. So that means overall. We now have to question 2.5. And figure 3 shows a diagram of 4 cells, A, B, C, and D. And each cell is surrounded by carbon dioxide molecules. So these black dots represent carbon dioxide molecules. You can also see that from the key here. And 2.5 is asking us in which cell will carbon dioxide move into at the fastest rate? And we need to give a reason for your answer and to tick 1 box. So you're going to get 1 mark for the ticking of the box and the reason. Now, this is all to do with the difference in concentration outside and inside the cell. So what we're looking for is the biggest difference in concentration. And the difference in concentration is referred to as the concentration gradient. So what we need here is what would be the biggest concentration gradient, the biggest difference in concentration outside and inside the cell. So looking at these, we can see it is cell A. There's a higher concentration here and low here. It's the biggest difference. Okay, so for example in B, there'd be no concentration gradient, so diffusion wouldn't be happening. There'll be no net movement of particles. So putting our reason now, we could say the reason therefore is the largest concentration gradient is in cell A. But just be very, very clear about saying exactly which cell. So question 2.6. A student investigated the effect of different colors of light on the rate of photosynthesis, and figure 4, which we can see here, shows the apparatus the student used, so the equipment the student used. And the student placed the apparatus in blue light, then in green light, and then in red light. And then the student measured the rate of photosynthesis in each color of light. Now, whenever I see a practical, I always think to myself, what are the independent, dependent, and control variables? So the independent variable is the variable that we measure, and the independent variable here is the color of light. Okay, so the light color. And the dependent variable is the variable that we measure, and we can see down here, it is the rate of photosynthesis. So that's being measured. It's always good to recognize this, because later in the scheme of questioning, they could ask you those questions anyway. So it's good to have an idea in your head. It really helps understanding exactly what's going on in the practical if you know your variables. And also let's think about some control variables, some variables that we're keeping the same. Control variables, I'll write them over here. Could be the species of pondweed. Okay, so the species of pondweed, the pondweed's mass. And also here, you could say the temperature, because temperature can affect the rate of photosynthesis. And you can also say carbon dioxide concentration as well as control variables. Now, let's go to question 2.6 specifically. And it's asking, what two measurements should the student make to calculate the rate of photosynthesis? So the rate, how fast it's happening. There's a couple of things you could do here. Obviously, sorry, these bubbles of gas are going to be oxygen. Now, what you could do is you could count the number of bubbles, and you could count the number of bubbles in a given time. So you may have done this practical in class where you counted the number of bubbles produced per minute. So giving any unit of time is absolutely fine here, but I just do per minute because most of you have probably done that. Now, there is a variation of this type of practical where you can use a gas syringe and measure the volume of gas produced per minute in 2 minutes, whatever unit time you propose. Now, continuing on with questions on this practical 2.7, and we have to give two variables that the student should keep the same in the investigation. Now, as I previously mentioned in question 2.6, they are the control variables, and you'll know that I've already named them, so let's just get straight into them. Let's do a few of them now. So temperature, specifically you could say the temperature of the water. Okay, try and be as specific as you possibly can. So you've got these colors of light. One of the control variables I didn't mention would be the light intensity. So you're changing the color of the light, but how much light is hitting the plant would be the light intensity. So that means you'd have the light, a specific distance away from. Now we've got a graph question for 2.8. And we've got to complete the graph, which is figure 5. And there's a nice checklist here, 1 mark per bullet point. So we've got to label the y-axis with a suitable scale, plot the data from table 2 as a bar chart. So this is the data from the experiment with the different colors of light and the rate of photosynthesis. And then we have to label each bar. So let's go through step by step. Let's actually, first of all, let's do a suitable scale. So we look at the results. Starting at 0, 9. So you want to go just above the highest value. So I'd go to 10. So I'd label like this. Now the label, you literally just copy what's in the table here. Independent variable is always on the left in the table. The dependent variable is always on the right. And the independent variable always goes on the x-axis of the graph and the dependent variable on the y. So I would label here rate of photosynthesis. Now, arbitrary units, if you ever see that, it's not a thing. It just means the units, for the sake of the question, don't matter. So just forget about it. Now, we can say here we have labeled the y-axis. Now we've given a suitable scale. And now we have to plot the data from table 2 as a bar chart and label each bar. So just for sake of ease, I'm just going to label each bar what it'll be here. So blue here, leave a space, green here. Each bar is going to be one large square, and leave a space, and then red here. And here we can see our bars. Okay, so 9, 1, and 8. Little exam tip for you. So now we've labelled each bar and we've plotted the data. So they're your 4 marks. You're allowed plus or minus half a small square. So there's a little bit of wiggle room for error, but try and be as exact as you possibly can to guarantee those marks. Question 2.9. Looking at table 2, again, our results from the practical, where we were changing the color of light, looking at the rate of photosynthesis. And the question is asking what color of light should be used to grow plants in a greenhouse. And we've got to tick one box. And if you're growing plants in a greenhouse, you want the fastest rate of photosynthesis possible. So therefore it is blue. Now on to question 3. Question 3, all about diseases. And rose black spot is a disease where black spots develop on the leaves of rose plants. And question 3.1 is asking us what type of pathogen causes rose black spot disease. And as per usual, we're ticking one box. First of all, it's worth knowing what a pathogen is. It is a disease causing microorganism. Okay. And this is just a simple knowledge question. And rose black spot is caused by a fungus. Question 3.2. Plants with rose black spot disease, remember caused by a fungus, often have yellow leaves. Suggest one reason why the leaves are yellow instead of green. Now, this we need to think, well, why is a plant green in the first place? Well, a plant is green, or bits of the plant are green, because it contains chlorophyll. And this is where our answer lies. So if the leaves are not green, they are now yellow, it will be because there is no chlorophyll. You could also say, make reference to there being no chloroplasts. That's where the chlorophyll is found as well if you wanted to. But the simplest answer there, if a plant is no longer green, there is no more chlorophyll. Question 3.3. We have to explain why plants with yellow leaves grow slowly. Remember, these questions are always linked. So 3.3 to 3.2 to 3.1. Remember, this leaf has rose black spot disease caused by a fungus, and there is no chlorophyll present. Hence why it's not green anymore. It is now yellow. So chlorophyll absorbs sunlight for photosynthesis. So if they're growing more slowly, it means very simply there's going to be less photosynthesis taking place. And less photosynthesis taking place means there's going to be less glucose produced, therefore there's going to be a slower growth. 3.4. The spread of rose black spot can be controlled using different methods. We need to draw one line from each method of control to the explanation of how it works. So the methods of control are on the left here and the explanations are on the right. So remove and burn infected leaves. We do that it will kill the pathogens. And our second method of control, water the roots of the plant only, not the leaves. And this reduces the chance of pathogens being spread by water droplets. Another multiple choice question here for 3.5. Tobacco plants may become infected with a pathogen called TMV. What type of pathogen is TMV? And we're ticking one box. So if you know what TMV stands for, this will tell you exactly what type of pathogen it is. And TMV stands for tobacco mosaic virus. Because when a tobacco plant is infected with TMV, it gives a mosaic pattern. And look, virus there. Therefore, it's a virus. 3.6. Now on to malaria, which is a disease caused by a protist. And the question's asking us, how is the malaria pathogen transferred to humans? Well, very simply, it is via mosquitoes, or you can say mosquito bites. So I'm going to say mosquito bites. Our last question of question 3.7, how can the spread of malaria pathogens be reduced? Tick in one box. So it's not a sexually transmitted disease. Cooking food thoroughly is not really going to make a difference here. It's transmitted via mosquitoes. 3.7 now, the last question of question 3. How can the spread of malaria pathogens be produced? And we've got to tick one box again. The answer here is to drain water from swamps. Mosquitoes, which we know transmit the pathogen, they'll leave their eggs in standing water. So that's water that's not flowing, as in, let's say, a river or a stream. So you remove the swamp water, you remove the eggs, and then you sort of stop the life cycle of the pathogen. Question number 4 now. Cigarette smoking is the main cause of cancer in the UK, and mutations in cells cause cancer. Now, 4.1 is asking us, where in a cell do mutations happen? And we've got to tick one box. Now, a mutation is the change in the genetic code of an organism. And that's the DNA that makes up the genetic code. And once you know that mutations happen to DNA, we can then go, well, where is DNA found in the cell? It's not in the cell membrane. It's not in the cytoplasm. It is in the nucleus. Question 4.2 now. And the question's asking us, why do some cancers develop into large tumours? And again, we've got to tick one box. First of all, with these questions, to get to the right answer, you need to know your definitions of the key terms. So let's define what cancer is. And cancer is uncontrolled cell division. And when cells divide uncontrollably, they can develop into sort of like a mass of cells, and that's what a tumour is. Now, once we know that definition, we can look at our options here, and it's clear the answer here is the first one. Cells never stop dividing. So I know it sometimes can be a bit monotonous, a bit boring, but learn your key terms. It makes interpreting answers. So question 4.3, cigarette smoking has been linked to many different types of cancer. Lung cancer is the most common type of cancer caused by smoking. And they're asking for us to suggest one reason why. Well, if you think about it, smoke is inhaled. So we inhale it, it's going to go straight down into our lungs. So that's really your answer, that smoke is inhaled. Okay, you could also say carcinogens are inhaled. They're chemicals that can cause cancer. Question 4.4 now. A person with lung cancer can develop secondary cancers in other parts of the body. We need to describe how this can happen. Now, secondary cancers, this is when we say the cancer is spread. That's known as a metastases, or when the cancer metastasizes. How can this happen? So it happens very simply. The cancer cells are transported via blood and then they end up lodged in other organs and parts of the body. Question 4.5 now, moving steadily on. Sometimes a person may need a lung transplant. That's where their lungs are removed, the patient's lung is removed, and somebody else's lung is transplanted in its place. The National Health Service, the NHS, will not offer a lung transplant to a person who smokes. Suggest one reason why. Now, again, there are plenty of things you could say about this. The obvious thing is the patient may also damage the new lung by smoking. Okay, it's going to be a waste of a lung. Okay, a healthy lung would otherwise be wasted. You could also say, especially these days, the NHS has limited amounts of money. If you said something like the NHS doesn't have enough money to provide lungs to people who smoke, that would also be a valid answer here. We've got a few questions now coming up that that ask questions based on figure 6. And that's figure 6, that's this graph here. And it shows data about skin cancer in males for different age groups in the UK. And the data shows the number of new cases of skin cancer in 1 year. Now, remember, whenever you see a graph or table, it's very good to really quickly just get an idea of the trends and patterns in that graph, because it's more likely than not that they're going to ask you a question about that. Now, remember, in graphs, the independent variable, what is being changed, is at the bottom. So the age is changing, okay, different ages. And what's being measured is the dependent variable, and that's the number of new cases of skin cancer in that 1 year. Let's look at the trends and patterns in this graph. You can see that as the age increases, the number of new cases in a year increases up to 65 to 69. And then after, from 65 to 69 to 90 plus, we see a decrease. Okay. So whenever you see a graph, very quickly in your head, just look at the trends and patterns, because more often than not, you're going to be asked about that later on in the line of questioning. So 4.6, which is based on this figure 6 that we've just been looking at, the question's asking how many more new cases of skin cancer are there in males aged 40 to 44 than males aged 15 to 19. So here's 15 to 19, here's 40 to 44. So to find out how many more, we've got to know the values for both of them, so how many new cases they were in this particular year. To do that, you need to know the value of one small square. There's a nice little trick to know the value of a small square on any graph. It is equal to the difference between the values divided by the number of small squares. So let me explain that. The difference between the values as we go up the graph here is 100, and the number of small squares is 5. So if you do 100 divided by 5, the answer is going to be 20 cases. So each small square represents 20 cases. So you can see 15 to 19, one small square. So we have 20 cases. And if we go along to 40 to 44, you can see here it's 300 plus one small square, it's 320 cases. So 320 minus 20 equals 300 more new cases. Okay, remember that little trick, you can work out the value of a small square on any graph. Question 4.7. There are no new cases of skin cancer diagnosed in males younger than 15 years of age. Suggest one reason why. This is very low at 15 to 19, but there's no new cases younger than 15, hence why it's not included on the graph. Think about it. Under 15 years of age, they're very young. They would have very little DNA damage. Okay, you could also say they haven't really been exposed to UV light very much in comparison to people who are in their 50s and 60s. You could also say that people who are younger, they tend to use suntan cream a lot more than some older people. So that's another possible answer you could give there. But they've had little DNA damage in comparison is the most obvious answer. Because remember, that DNA damage leads to mutation, which can lead to cancer. On to question 4.8. And they're asking us to give one conclusion from the data in figure 6. When you ask for a conclusion, they're asking for you to interpret the trends and patterns. Remember, we did that a little bit earlier in the video. Okay. There are a few conclusions we can make, but they're based on the trends and patterns that we described earlier. You could say from ages from 15 to 19 to 65 to 69, there was an increase in the number of new cases per year. You could also say from the ages of 65 to 69 to 90 plus years old, there was a decrease in the number of new cases of skin cancer per year. Next up is question 4.9. Survival rates for all types of cancers have improved over the last 20 years. And we've got to suggest 2 reasons why that is. Now, thankfully, this is actually quite some positive news, makes a change from what's in the news these days. So why have survival rates improved? Well, very simply, we have improved treatments. So that would be things like drugs. You can say improved drugs or new drugs. And you could also say as well, due to cancer screening, you get sort of improved diagnosis. So improved or earlier diagnosis, and that is via cancer screening. So an example of this would be, males over 50 have to have a prostate exam, and I think they go yearly, and that's to check for any signs of prostate cancer, which is quite common in males of that age. And also a lot of women go for cervical cancer screens called smear tests, and they go for those yearly, again, once they get to a certain age. And this has really improved the earlier diagnosis, and the earlier you diagnose these conditions, the quicker you can treat them. Question 5 now. Bacteria can cause a variety of diseases in humans. And what are 2 similarities between a bacterial cell and an animal cell? And this time we've got to tick 2 boxes. So here you have to know the different cell types and also the organelles that make them up, what they share and what they do not share. So both have a cell membrane. Yes, they do. Both have a cell wall. That's not true. Only bacterial cells have a cell wall. Both have a nucleus. Again, that's not true. Only animal cells have a nucleus. Both have a cytoplasm. That is true. And both have plasmids. That is not true. Only bacterial cells have plasmids. They're small circular pieces of DNA that float in the cytoplasm. Question 5.2. Salmonella food poisoning is caused by bacteria in food. We need to give one symptom of salmonella food poisoning. Remember, a symptom, just make sure we're defining these keywords, is a sign given off by the body that something is not right. It's a sign of illness. And do not refer to vomiting or diarrhea in your answer. Well, with vomiting and diarrhea being ignored, answers here would be a fever or stomach cramps. They're also signs, therefore symptoms, of salmonella food poisoning. Question 5.3 now. And the question is asking, what is the name of the first antibiotic developed? Well, simple recall question here. If you know it, you know it. It is called penicillin. And it was first discovered by a scientist based in London called Alexander Fleming. A little bit of background there for you. So now we have some questions based on figure 7, which we can see here. And it shows the concentration of live bacteria in a child's body, how it changed when taking a course of antibiotics. So again, our independent variable here is the time in days, and our dependent variable is the concentration of live bacteria in the body. So again, before we get into the series of questions, let's just briefly describe the trends and patterns that we can see. So the course of antibiotics is started here. For around a day, we see an increase in a number of bacteria. Then after 1 day, we see a steady decrease down to 0 bacteria just after 4.5 days, give or take. So again, just to re-emphasize, whenever you see a graph, always in your head, just go through and see if you can describe the trend and pattern. It will help you with the later questions. Question 5.4 then. The concentration of live bacteria in the body continued to increase after starting the course of antibiotics. So in the previous segment where I described figure 7, it's here. This increase despite the course of antibiotics having been started. And they would like us in question 5.4 to suggest a reason why we've seen that increase despite having started a course of antibiotics that should kill bacteria. So the obvious reason here is just because you started a course of antibiotics, it doesn't mean enough time has passed to actually kill them. So what there is here, there is a time delay prior to the antibiotic killing the bacteria. You could also give an answer that makes reference to the bacteria reproducing still as the course of antibiotics has started. So if you said the bacteria were still reproducing, that would be fine because they haven't yet been killed still. Let's now read through question 5.5. After 3 days of taking the antibiotic, the child felt better and there was still bacteria in the child's body. Why did the child feel better? We have to tick one box. And our possible answers: the bacteria had become immune to the antibiotic. The child had become resistant to the bacteria. There were fewer toxins in the body than at day 0. Now, background knowledge here: bacteria, they produce toxins which are poisons. And it's the poisons, the toxins that make us feel ill. They give us the symptoms of feeling ill. So our answer is the third one. There were fewer toxins in the body than at day 0. All of them hadn't been killed yet, but the child felt better. Less toxins. Question 5.6. And they're asking us to suggest why doctors do not give antibiotics to patients with minor infections. Now, first thing we could say, well, the first thing that springs to mind for me is that not all infections are caused by bacteria. Remember, antibiotics only work on bacteria. So we could say something along the lines of some infections are caused by viruses. So the antibiotics wouldn't work anyway. Also, doctors need to be very mindful that what if they keep giving antibiotics to every patient with even a minor bacterial infection, what it can lead to is the bacteria becoming resistant to that antibiotic. So the antibiotic will not be able to kill that bacteria. And we're already seeing strains of bacteria appear in the population that are not killed by certain antibiotics. And again, that could be very dangerous in the long term. Question 5.7. And we have figure 8 here, and it shows blood being viewed using a microscope. And the question's asking: a vaccine will stimulate the production of antibodies. Which part of the blood in figure 8 produces antibodies? Well, first, it's good for your revision, going through these walkthroughs, that you know what a vaccine is. I'm always stressing the importance of knowing your keywords. A vaccine is an injection, and not all injections, but a lot are, of a dead or inactive form of a pathogen. And by having that injection, your body then produces antibodies. So when you're reinfected with the real live pathogen, the antibodies destroy it and you don't get infected. So, or you don't get sick, really severely sick. Now, to know which part of the blood produces antibodies, yes, you could just know the answer, but also it's nice to be able to know them from their structures here as well. So A is a red blood cell, B is a white blood cell, C is the plasma, which is a straw-coloured liquid which transports nutrients such as digestion products like glucose and also that's red blood cells, carbon dioxide. and D are platelets. They're cell fragments that help the blood clot. So antibodies, white blood cells. Our answer then is B. Question 5.8 now. Which part of the blood in figure 8 starts the clotting process? We have to tick one box. Now remember, the clotting process, that was the platelets, and they were D. So D is the answer there. Okay, question number 6. And these questions are about cell division. 6.1. We have to write the biological structures from the box in the correct order of size. And we can go here from smallest to largest. You can go from largest to smallest, but I'm just going to follow the question. Well, everything is made of atoms, right? And atoms make molecules, such as, in this case, we're looking at cells, so DNA. DNA is the molecule. A short sequence of DNA, a short section of DNA is a gene. So that would make genes the smallest. Again, it's all about knowing your definitions. Okay. And multiple genes, they make up a chromosome. A chromosome is one extremely long molecule of DNA made up of genes. So that would put the chromosome there as our next one. And then, so when we've done that, it's probably good to cross them off as well so we know. Then chromosomes, where are chromosomes found? They're found inside the nucleus, along with genes, right? Because genes are found on chromosomes. And then the nucleus is in the cell. So gene, chromosome, nucleus, cell. Question 6.2 now, and we've got to look at figure 9. That's here. And figure 9 shows how a fertilized egg cell can produce specialized cells. So we have our fertilized egg cell here. That's also known as a zygote. It's always good to know. So we get that fertilized egg cell when sperm and egg cell nuclei fuse, then undergoes cell division. So in this case, it will be mitosis, the cell division, and that will lead to a ball of cells known as an embryo. Then we have process A. Now 6.2 is asking us to name process A, and we go from an embryo to red blood cells, muscle cells, nerve cells. Now, all of these cells have very specific functions, so we call these specialized cells. So they're cells that are adapted to have a particular function in the body. Whereas these cells in the embryo, these will be stem cells where they haven't yet become specialized. So what's the process that leads to these specialized cells? Well, it's known as cell differentiation. Question 6.3 now. How many cell divisions are needed to form a 16-cell embryo from the original fertilized egg cell? So it's how many cell divisions does it take to get from one cell to 16 cells? Now remember, I said the cell division was mitosis, so what we end up with is one cell dividing to become 2 cells. Okay, so each cell produces 2 cells. Now mathematically, if we thought it was one division, it would be 2 to the power of 1. If we thought it was 2 divisions, it would be 2 to the power of 2, which we know is 2 times 2 equals 4. Okay, 2 to the power of 3 divisions, which is 2 times 2 times 2. 2 times 2 is 4 times 2 is 8. Or if it was 4 divisions, it would be 2 times 2 times 2 times 2, which is 2 times 2 is 4 times 2 is 8 times 2 is 16. So that's our number. So therefore, it's 4 cell divisions. Now, if you're not comfortable using the standard form way of doing this, you could, if you wanted to, just draw it out. 1 becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16, but it can get a little bit messy, but you would always get to the right answer doing it that way. Question 6.4 now. In humans, a fertilized egg cell, remember that's our zygote, contains 23 pairs of chromosomes. So that means each of the 23 chromosomes, there are 2 of them, so we can also say that there are 46 chromosomes inside a fertilized egg cell. And question 6.4 is asking us how many chromosomes will there be in each of the embryo cells. Now, remember, I said that our zygote undergoes mitosis, a few rounds of mitosis, to end up as a ball of cells known as an embryo. So mitosis produces genetically identical daughter cells. Therefore, the embryo cells are also going to have 23 pairs of chromosomes, or 46 chromosomes. So here you could write 46, or you could write 23 pairs, and it'll be exactly right. Now, the only cells, so all body cells will have 46 chromosomes. However, sperm cells and egg cells will have 23 chromosomes. Okay, 23 chromosomes. That's one set. So what we call these cells that have one set, these are called haploid cells, and 23 is the haploid number for humans. When there's two sets of each chromosome, so the chromosomes are in pairs, these are known as diploid cells. So for humans, the diploid number is 46. Okay, 6.5, figure 10, that's down here, represents a cell cycle for a human embryonic cell. All cells all body cells go through a similar cycle, and we need to describe one change in the cell that occurs during each of the stages of the cell cycle. So stage 1, this is the stage that a cell is in most of the time. And in this stage, there's a few things we could say here, but the most common one is the DNA replicates. There's also cell growth. So the cell's growing, the DNA is replicating. And you could also say there's an increase in the number of organelles as well at this stage. So there's 3 different ways of saying, describing what's happening. Stage 2 is mitosis. So you've got to sort of say, you can't just copy that and write mitosis there. You've got to say, well, what are key things that are happening during mitosis? And one of them is that 2 nuclei form. Okay. You could also say something like the chromosomes lining up in the center of the cell, and then one set of chromosomes get pulled to one side of the cell, and the other set of chromosomes get pulled to the other side of the cell, to opposite sides of the cell. But 2 nuclei are forming during mitosis. And lastly, stage 3, this is where we end up at this point with 2 genetically identical daughter cells. So in the end, we've had the one cell, they've undergone mitosis, and by stage 3, we have our 2 genetically identical daughter cells that are formed, and that there is stage 3. So here we are at figure 11 in question 6.6. This is the last question on the paper now. And figure 11 shows the mean height of boys and girls from birth to age 18. And we can see here the dashed line, boys, and the bold line, girls. Now, I always say, identify your variables. On the X, we always have the independent variable, so the age in years is changing. And the dependent variable, so what's being measured, is the mean height in centimeters. So before we get on to the actual question, I want you, whenever you see graphs and data like this, to think about the trends and patterns. Okay, so what we can see here is both of their heights are increasing over time, which you'd expect in living organisms that they grow and they get taller. But we can see a few little deviations in the graph that are worth explaining here. So at birth, you can see that's over here, boys are slightly taller than girls, and boys are slightly taller than girls all the way up to this point here, which, look on the graph, about 11 years old. And girls are then taller than boys up to about age 14. And then after that, girls sort of around sort of 14 years old, they tend to stop growing. Whereas boys, they carry on growing up until about the age of 18. And overall, you can see here, boys are taller than girls. So the question here, the 6 marker, we have to compare the growth of boys and the growth of girls, and we have to use data from figure 11 in your answer. So me just going through them, describing the trends and patterns is going to come in really handy here, but we do need to use some data as well in our answer. So let's make, first of all, just some comparisons first of all, and then we'll use data. So with 6 marks, it's always good to split up your answer. So let's make some comparisons. Okay, so here are some general comparisons. The height of boys at birth is slightly taller than girls. You can see that here. The rate of growth is similar up to around age 11. It's not identical, but it's similar. It's following the same sort of rate. That's the amount of growth you would see per year. Very similar because the line is a similar steepness. And by 18, boys are taller than girls. Now, we want to use some data to back up our answer here. And let's use our data to compare the heights. So let's go from here. So we can say at 18 is 180 centimeters. So that's just under 6 foot. Whereas girls there, it's about 165. So girls' height is 165 centimeters. So we've made some comparative statements here, and we have also used data that will ensure we get the 6 marks. Just to be safe though, I might just make one more comparison on here, actually, just one more. Talk about this area of the graph here, and we can say between the ages of 11 and 14. So from 11 to 14, girls are taller than boys, just to ensure we get that maximum of 6 marks. And to get 6 marks, you have to at some point reference the data, and that's what I've done here. Last little part of question 6 now, and this is right at the end of the paper. Last question here. If you've made it this far, well done. We need to just give one way that cell division by mitosis is important in fully grown animals. So you can't say growth here because the animal is fully grown. So how is mitosis important once an organism is fully grown? So it's all about repair here. So repair of tissues, or you could also say replacement of cells. Remember, you can't say anything about growth here. And that's the end of the paper. Well done, guys. I'll see you in the next one.